Hello, my name is Emily Diaz and I'm a QBIC sophomore.
And my name is Gabriela Padilla and I'm a QBIC junior.
We are currently a part of Dr. Rodriguez-Lanetti's UBIOS lab.
The goal of our lab is to come up with a solution to the ongoing coral bleaching problem around the world. Due to the warming ocean temperatures, the symbionts that live in the corals leave them, causing bleaching and death.
In our lab, we analyze the symbiotic relationship between the corals and the symbionts by using our model organism, anemones. We experiment with anemones and different species of symbionts in various nutrient treatments.
We're trying to discover how these nutrients may affect the health of the symbiosis. We hope that these treatments may be the answer to solving the coral bleaching problem.
Now to Justin and Ivan that will give you a quick tour of our lab.
Hi everybody, I'm Justin Prats.
And I'm Ivan Garces.
And welcome to the
UBIOS lab. Over here we have our incubator where we keep our two species of our model organism. We have our Lanuca on the left-hand side and the Minutum on the right-hand side to keep them separate. So over here for the Lanuca, we have them separated by treatments. And as well on the right-hand side for the Minutum, we also have them separated by treatment.
We have many useful instruments in this lab, but one of my personal favorites is the vortexer. We can take our anemone samples and convert them into a usable form. It's a very simple but effective tool. So once we break down our anemones, we can analyze them, their DNA or their proteins using blotting, or we can even centrifuge them to separate the symbiont and host components.
An important part of this lab is husbandry. So what we do is we come in here throughout the week and we clean these cups that have the anemones inside them. So the anemones by themselves produce waste in these cups, and what we do is we come in and clean them to ensure that they're in a clean environment. So looking at this cup, this is all the waste produced by the anemone throughout the week, and what we do is we come in here and we clean them to make sure they look like this and remove all the waste, and then we fill them up with their corresponding treatments.
Hi, my name is Andy Garcia, and I work at Dr. Allen's Neurocircuitry and Cognition Lab. At the lab, we investigate the circuit connecting the hippocampus and the medial prefrontal cortex. This circuit is abnormal in diseases such as schizophrenia and Alzheimer's, which means that a better understanding of it will lead to better diagnostic and therapeutic tools in clinical cases where these diseases are apparent. I work with graduate student Manasa Yagandran in investigating the neurobiology of time in rats using a sequence memory task. With the help of electrophysiology and optogenetics, we're able to gain a better understanding as to how this circuit is needed for the memory of sequences. And we're doing this by both seeing and manipulating neural activity during the task. I'm also independently assessing the synchrony between the medial prefrontal cortex and the hippocampus. And in doing so, I'm trying to find the degree to which these two regions act as a team in order for us to both recall memory and execute the corresponding behavior. We hope that by using rats as a model, which share much of the same biology, we can get better access to diagnostic and therapeutic tools in medicine.
Hi everyone, my name is Daniel Cambron, and I'm a senior majoring in biological sciences. As a QBIC scholar and a Mark U-Star affiliate, I've had the opportunity to conduct research in a variety of labs. Right now, I'm doing my undergraduate thesis in Dr. Hutchison's cardiovascular matrix remodeling lab through the Department of Biomedical Engineering. I started conducting research in this lab back in the fall of 2019, and my experience has been nothing but amazing. Dr. Hutchison has been one of the best mentors I've had the privilege of working with, and has helped me become the scientist I am today. The lab has two general focuses, valvular and vascular. The valvular team performs experiments to gain more knowledge of how the valve functions during diseases. The other side of the lab focuses on the vascular aspect of diseases, like the buildup of plaque in arteries. This is where my project falls under. I study the role of dynamic environments in vesicle trafficking. These vesicles of interest are little containers that are released from cells and travel to damaged areas in the artery. The vesicles are important because they contain some of the proteins required for calcifications to form, which leads to heart attacks and strokes. My project aims to better understand how these cells release the vesicles in response to changes in the environment. One of the coolest things I get to do in the lab is stretch pig cells. After treating the cells with inhibitors, I stretch the cells in a silicone dish that mimics the stretch felt by cells in the body. By measuring the protein levels and using microscopes, we have learned how the cells release different amounts of vesicles depending on the type of stretch they feel. We have also started to understand what proteins are responsible for the release of these vesicles. Thank you all for listening, and I hope you enjoyed the talk. Proud to be an FIU Panther.
Hello everyone, my name is Melanie Silva and I am a proud research assistant at Dr. Lorraine E. Barrick's Infant Development Lab. I applied for this lab back in December of 2019 and it has been one of the best decisions I have made in college. Amidst a pandemic, I am honored to say this lab is up and running, completing procedures remotely, and I am beyond lucky to be a part of it. Ultimately, the Infant Development Lab strives to better comprehend infants' perception of events and objects in their surroundings, and how early emerging skills such as attention and matching sights and sounds of speech and object events lead to more complex skills such as language and social functioning later in childhood. As a research assistant, I am expected to participate in weekly lab meetings, participate in lab activities 12 hours a week, including recruiting and welcoming participants, as well as collecting, coding, and entering data. Due to COVID-19, these tasks have been slightly changed. Currently, videos of parent-child interactions are being coded through an application called DataVoo. I participate in transcribing the language between the parent and the child for later analysis. Usually, these interactions would be observed in our ideal lab setting with the mom or dad and child sitting across from each other at a table, with toys provided. They would be instructed to play normally as they would at home for a certain period of time while they are being recorded. However, because of the pandemic, the parent-child interactions take place in their home and are recorded through Zoom. They are given the same instructions with the slight alteration that they must use toys at home, and their device camera must be oriented at a certain angle to properly view the two individuals. These are one of the many methods the Infant Development Lab has changed to fit remote needs. I'm truly in awe of how well it has been executed. Over the years, results of studies in the lab have demonstrated that at 3 to 6 months of age, infants can detect a variety of properties of speech, including rhythm, tempo, prosody, and face-voice synchrony. Infants can distinguish between sexes, adult and children faces, and recognize which voices and faces belong together. In addition, they can remember events and everyday actions, with this recollection persisting across months. Overall, this lab has charted the typical development of a variety of foundational skills in infancy and early childhood that can be used to identify children at risk for atypical development. I am forever grateful that I was offered a position as a research assistant.
Hi, my name is Melanie Perez, and I'm current QBIC sophomore working in Dr. Dwan's lab in FIU School of Medicine. Dr. Dwan is currently pursuing multiple projects pertaining to lung cancer and the TP53 gene, which produces a protein called P53 that regulates the cell cycle as well as apoptosis and acts as a tumor suppressor. However, I'm currently working on a project about expressions of microRNA within lung cancer cells and non-tumor lung cells in transgenic mice. Using nanostring technology, we are able to identify which microRNA is being under and overexpressed in each of these situations. Knowing which microRNA is being under or overexpressed, we are able to identify the role of P53 in the development of lung cancer. Thank you and stay safe.
Hi, my name is Nicole Diaz and I am currently a senior undergraduate student at FIU. I am a QBIC scholar as well as a Mark U-Star Research Fellow. Over the summer, I had the opportunity to work in Dr. Stern's lab at Johns Hopkins University via a remote research experience. This was my first time working in an oncology lab where we specifically looked at the underlying impacts of racial and ethnic differences among breast cancer patients. Currently, I am in Dr. Liu's lab at FIU. This is a biochemistry lab that specifically focuses on DNA damage and how we can find a treatment or therapy for some neurodegenerative diseases. And following graduation, I intend to get a PhD degree in life sciences, and I appreciate the QBIC program and the Mark U-Star program for their continuous support and guidance throughout my undergraduate career.
Hello, my name is Pablo Puente. I am a QBIC scholar. I am a senior, and I just wanted to talk to you about Images Lab, led by Dr. Rodriguez Lanetti. And with my mentor, Daniel Marcellus, we have been able to work through this whole pandemic. With COVID-19, it has been very difficult to go into the lab and actually get experimental data. And it has been a very difficult situation to be put a lot of researchers in, but that doesn't mean it is less enlightening or less rewarding at the end. Once you actually read the literature about something that you're actually passionate about, it feels very rewarding. So I am in the Arch program, also in the Honors College with a proposal track. So I do have research to do. And one of the main parts of the research is actually background information and literature reviews. So I actually have to research a lot of articles and actually come up with background that will actually help support my main points, my research questions, my research aims. So this is a very important process. So I just wanted to explain a little bit about the methodology that I use in my lab. I do a lot of observational data with animal behavior and Exoptasia pallida, which is a type of anemone. So I try to look at aposymbiotic anemones and symbiotic anemones with Raviolum minudum and Cymbidium lanucci. So I chose this background because it is actually one of the main aims of the lab is to find heat tolerant species within, because as you all may know, there's many climatic events that are destroying these beautiful ecosystems for the coral reefs. So there needs to be a lot of research. There's a lot of research to be done with this ecosystem to help it from decaying, help from bleaching events to keep happening as often as they do. So I just wanted to share a little bit of the data that I have gotten throughout my years of the lab. There's some of the data that I have gotten. This is observational for digestion. So this is very good example of what I do in my lab. I try to look at patterns, see trends between aposymbiotic and symbiotic relations. Also, I will try to show you how I have other charts that I come up with for phototaxis. Phototaxis is moving towards light. That is another pattern that I have seen in my lab. So these are very rewarding studies that I have done before. And now due to the COVID-19, I haven't been able to get back into the lab. But once again, just knowing that once you get back in, you can do a lot of research, and there's a lot of research to be done for the common cause, which is try to help these species from dying out. Thank you for listening.